Hello everyone, I'm Sidi from Ben-Gurion University of the Negev in Israel. I'll present my poster, Redundancy-Weighting the PDB for Detailed Secondary Structure Prediction. I would like to thank the organizers for this opportunity to present our study. In this presentation, I will promote two concepts. One, we should embrace the PDB redundancy instead of fighting it. And two, the detailed secondary structure prediction is both feasible and useful. Downsampling to non-redundant subsets is the common method for dealing with the biases in the PDB. Non-redundant datasets, which comprise of cluster representatives, are presumably unbiased, yet about 75% of the data is lost, and all the diversity within clusters is ignored. We offer an alternative approach, called redundancy weighting, which downweights the redundant entries instead of removing them. This way, much of the data is preserved, yet the bias problem is alleviated. Methods for secondary structure prediction map each residue in a protein sequence to a secondary structure class. Current methods predict either 3 or 8 classes. In 2003, Karchin and co-workers proposed 13 classes classification, called STR2, which splits the E class of DSSP into 6 subclasses based on their orientation and position. Here, we apply the redundancy weighting approach to the problem of secondary structure prediction with 8 and 13 classes, and show that it improves performance. We consider the PDB as a graph, where the chains are nodes and edges are similarities. To evaluate performance, we partition the graph to 3 disjoint parts: training, validation, and test. Then, we choose cluster representatives for each subset. So we have non-redundant test set, non-redundant validation set, and for the training, we have a redundant training set and a subset of cluster representatives, which is a non-redundant training set. We employ per residue weighting. Each chain we align with its neighbors according to the similarity graph, and each residue weight is the inverse number of identities in the alignments. We took the deep learning models from Drori et al. And modified them to support weighted samples. In order to determine the number of epochs for each model and dataset, we used an early stopping mechanism that stops training when the accuracy on the validation reaches a plateau. In the plot, we see the validation and training accuracies of 2 models, one trained on the non-redundant training set in green and orange, and the second on the redundancy-weighted training set in red and blue. The redundancy-weighted dataset is larger and thus require fewer epochs. We tested the performance of the trained deep learning models on the non-redundant test set. The table presents the results on one of the partitions we made. The performance is measured by Q-measure and Matthews correlation coefficient. We emphasize the best performance for each model and measure in a bold font. Consistently, it is the models trained on the redundancy-weighted datasets. Note that the ensemble average model is the best performing overall and on each dataset. To evaluate the utility of detailed secondary structure prediction, we incorporated features derived from 8 and 13 classes predictions into our EMA method, MeshiScore. The table presents the EMA results on the 3 modeling targets of CASP13. It shows that using features derived from STR2 prediction lead to better performances. The same trend, though less pronounced, appears on the whole CASP13 decoy set. To summarize, most importantly, we show that training on redundancy-weighted datasets improves the performances of secondary structure prediction methods. We believe that this is also true for other aspects of computational structural biology. Additionally, we revived the STR2 alphabet for secondary structure prediction and showed that it is useful for estimation of protein model accuracy. Finally, our easy-to-use JSON datasets are publicly available, and we hope they will encourage more efforts in this direction. Thank you for listening. I would be happy to answer your questions.